Hi everyone, and welcome to our video on some exam questions related to biological molecules. In specific, we are going to be focusing on lipids and carbohydrates. Our first question is from the 2016 AQA AS Biology Paper 1 and states, describe the difference between the structure of a triglyceride molecule and the structure of a phospholipid molecule. So this is a simple recall question. It doesn't require any application. We are just comparing between a triglyceride and a phospholipid. And the key difference is that a phospholipid has a phosphate group which replaces one of the fatty acid chains. If I show you the mock scheme for this question, it states just that. In phospholipid, one fatty acid replaced by a phosphate. And that's what you need to say in your answer. It says ignore references to saturated and unsaturated. As it does say in the question, you need to compare between the structure. And the key difference that they're looking for is the phosphate group. The second question is describe how you would test for the presence of a lipid in a sample of food. So again, over here, this is a common exam question and the examiner is looking for you to write down the sequence of steps, okay? And that's what all the mock schemes have in common. So to test for the presence of a lipid, well, you need to add ethanol, then water, and you need to say what would happen for a positive result. Well, for a positive result, I've just written it an add sign just for short. It would be a white emulsion. Okay, again, if I show you the mock scheme for this question, it says add ethanol, then add water, and it says a white emulsion for a positive result. Okay, it says reject ethanol or ethanol. You need the correct spelling of ethanol. It says accept alcohol or named alcohol. Accept milky, ignore cloudy. So you can't say cloudy anymore. You have to say either milky or emulsion, a white emulsion, and the sequence must be correct. Okay. If we move on to our next question, animal fats contain triglycerides with a high proportion of saturated fatty acids. If people have too much fat in their diet, absorption of the products of fat digestion can increase the risk of obesity. To help people lose weight, fat substitutes can be used to replace triglycerides in foods. Describe how a saturated fatty acid is different from an unsaturated fatty acid. Again, this should be a nice recall question and it's an easy 1 mark to get. What you need to say is that saturated fatty acids what do they contain? Well, saturated fatty acids, they contain only C to C single bonds, okay? And that will get you the mark. If I show you the mark scheme for this question, it says saturated contains single / no double bond between carbons. Okay, now instead of between carbons, you could have said R groups or variable groups. Okay, or you could have said the opposite of that, saying that unsaturated has at least one double bond between the carbons. Okay, but for saying this answer, we would have got 1 mark. Next question, figure 1 shows the structure of a fat substitute. The fat substitute cannot be digested in the gut by lipase, suggest why. So remember, this is a fat substitute. So remember, lipase is an enzyme, okay? And it will have an active site which is complementary in shape to its substrate. Now, if we use a fat substitute, that will have a different shaped substrate. Because it has a different shaped substrate, it won't be complementary to the active site of the lipase enzyme. Okay, and therefore, as with any enzymes question, the enzyme substrate complexes cannot form. Okay, so if I show you the mark scheme for this question, well, it says fat substitute is a different slash wrong shape slash not complementary. I would have gone for that, not complementary. And then the second mark is that it's unable to fit slash bind to the active site of the lipase or no enzyme substrate complex formed. I would have gone for that because normally with every enzymes questions, enzyme substrate complex stating that keyword would get you a mark. Okay, so I would have gone for not complementary, therefore enzyme substrate complexes cannot form. Okay, and if you state a bond, for example a peptide bond or a glycosidic bond, and it's the wrong name, just like that, then you will lose a mark. You will say penalized once. Okay, you have to say ester bond. That's the correct name of the bond. Now the second part of this question says this fat substitute is a lipid. Despite being a lipid, it cannot cross the cell surface membranes of the cells lining the gut. Suggest why it cannot cross the cell surface membranes. Now this question, it's about applying what we already know. If it's a lipid, it's a fat substitute, but it cannot cross the cell surface membrane. Now, if you link some knowledge of cell membranes into this, and it might not be able to cross because it's not lipid soluble, well, it is because it's a lipid. There has to be another reason. And that other reason is that it could be too large. Okay, it doesn't tell you the size of the substitute, so it could just be too large. Okay, or it could be polar, we don't know the charge of it, anything like that. So if I show you the mark scheme for this, it could say that it's hydrophilic, it's polar, it's too large, or it's too big. And it does say it's not lipid soluble because it is a lipid. Okay, so you have to say one of these reasons, and I would just have gone for that it is too large. Okay, next question. This is from the 2016 AQA A-level biology paper 1. Lipase is an enzyme that hydrolyzes triglycerides. A student investigated the hydrolysis of triglycerides in milk by human lipase at 20 degrees. He recorded the pH of a sample of milk before and after adding lipase. He used a pH meter to record pH, and we're given the graph here. Now it says suggest one advantage of using a pH meter rather than a pH indicator in this experiment. Now this is not explicitly covered on the spec, however it does come up in exam questions quite a lot. Okay, and whenever it's asking for the advantage, you need to look at the accuracy. Okay, pH meter will give you a more quantitative result. Okay, and a pH indicator will give you a color. Now the key thing with this is that a pH meter will give you greater accuracy, because if we used a pH indicator, that will give us a color, and color can be read by different people in different ways. So we would say that color change is subjective. Okay, so simply for this question, I would just write it down here, that a pH meter has greater accuracy. And because it has greater accuracy, why are we going to be using it? Is because the other pH indicator provides you a color, and color change is subjective. Okay, so color change is subjective. Now if I show you the mark scheme for this question, the mark scheme is now on numerical readings not subjective, color change subjective, gives quantitative data not qualitative, gives continuous data. You would have had any of those for 1 mark. And the second mark, you have to say why you would use it. Okay, so if it's not subjective, or if color change is subjective, or gives you quantitative data, why are you going to be using that set of data over the color change? Well, it's because that it's of greater accuracy. Okay, and it does say that you can state greater precision. Okay, so anything like that would get you the mark. Okay, next part of the question, it says explain why the pH decreases when the lipase is added to the milk. Well, if we go back to the question, we are investigating the hydrolysis of triglycerides. So when triglycerides are hydrolyzed, basically what happens is that the fatty acid chains are released. Okay, so why does the pH decrease? Well, when the lipase enzyme is added, the fatty acid chains are going to be released into the sample, and because they are released, remember it's fatty acid, because it's an acid, it's going to decrease the pH. Okay, so for this question, all I would say is that fatty acids have been produced, which will lower the pH. Okay, and the mark scheme wants just that. Fatty acids are produced and they will lower the pH. Now the next part of the question says, suggest why the pH remained constant after 2 minutes. Okay, if we go back to the graph here, after 2 minutes, around this point here, the pH remained roughly constant. Okay, and the question is asking, well, why is this? Well, the pH has remained constant, first of all, because no more fatty acids have been produced. Okay, no more fatty acids have been produced. And why is that? Maybe. Okay, why is that? Well, that is because perhaps all the triglycerides have been used up. Okay, no more fatty acids can be produced because all the triglycerides have been used up. Okay, and that's the most sensible answer. Okay, so I would say that no more fatty acids produced, and because if we go back here, the line is strictly horizontal, it doesn't sort of slowly decrease, it's strictly horizontal. That tells us that all the triglycerides have been used up. No more fatty acids produced, as all the triglycerides have been used up. Okay, that's what I would say. If we have a look at the mark scheme, it says no more fatty acids produced, all triglycerides slash fats slash lipids slash substrate used up, or enzyme is denatured. Okay, so I would have got both marks in that case. Now the final part of this question states the student carried out his experiment at 20 degrees. He then repeated the experiment at 15 degrees. Draw a line on the graph in figure 3 to show the results you would expect at 15 degrees. So this original experiment was carried out at 20 degrees. If it's carried out at 15 degrees, the reaction is going to happen a bit slower because you're at a lower temperature. However, you will start off from the same point and you will come to the same endpoint. Okay, so when drawing this line, start off with the same point and gradually come down and you will end at the same point, just like that. Okay, if I show you the mark scheme for this question, it says line starting at same point and falling above original line, levels off at same pH but later. Okay, and we would have got both marks there. Okay, next question is from the 2016 AQA AS Biology Paper 1, and this time it's related to carbohydrates. So it says scientists investigated the hydrolysis of sucrose in growing plant cells by an enzyme called SPS Name the products of the hydrolysis of sucrose. So again, this is a simple recall question. So, the products of hydrolysis of sucrose are glucose and fructose. Okay, easy 2 marks to get, and the mark scheme just says that glucose and fructose, it doesn't matter on the order that you have, and it says ignore alpha or beta glucose. Okay, now the next question is work out the rate and draw a graph. I won't draw the graph on here, but I will work out the rate. Okay, so it says the scientists grew plant cells in a culture for 12 days. At the start, there were only a few cells in the culture. Each day, they determined the mass of sucrose hydrolyzed by SPase in the plant cells in 1 hour. So we've got the days, and then we've got the mass of sucrose hydrolyzed by SPS in 1 hour. Okay, and we need to work out the rate of hydrolysis of sucrose by SPS. However, we need to work it out per minute. If this is per hour, to work out per minute, all we would do is divide these values by 60. Okay, because there are 60 minutes in an hour. So each of these values we would divide by 60. Okay, so the first one will get you 1.17 times 10 to the power of negative 3. The second one will get you 1.5 times 10 to the power of negative 3. The next one will get you 1.83 times 10 to the power of negative 3. The next one will get you 2.5 times 10 to the power of negative 3. The next one will be 3.33 times 10 to the power of negative 3. The next one will be 4 times 10 to the power of 3. And same for the last one. Okay, that's your rates. Now when you're plotting a graph, as you can see, all of these are times 10 to the power of negative 3. So we can just label our y-axis with the times 10 to the power of minus 3, and we can then label each of these big squares as just 1, 2, 3, and 4. Okay, it just makes it much easier. Okay, instead of working with really small values such as 0.00117. Okay, so it's better to have it in whole numbers and good numbers and label the y-axis as times 10 to the power of negative 3. So whatever value you have, you know that you are going to multiply by 10 to the power of negative 3 to get the actual answer. Okay, the day is going to go on the x-axis, and the rate of hydrolysis of sucrose is going to go on the y-axis. Remember your units and remember to label. Okay, if I show you the mark scheme for this, it says a line graph with the rate on the y-axis and day or time on the x-axis in linear scales. These are our answers, which we got over here. And it does say reject if 10 to the power of negative minus 3 on axis for each point. You should just label your axis with that, just like it's done at point 2 here. And the rates should be correctly plotted with the line connecting the points. Don't extrapolate, or you will lose the mark for that. Okay? Moving on to our final question. What can you conclude about the growth of the plant cells from these data? Explain how you reached your conclusion. Okay, so if we go back here, as you can see, if we look at our rate of hydrolysis of sucrose, as the days are increasing, we can see that the rate of hydrolysis of sucrose is also increasing. Okay, biggest jump over here, and then it levels off around this place. Okay, so that's all we need to describe and explain. So our conclusion will state the following: that sucrose hydrolysis is linked to some aspect of the growth, and the reason for that is that we can see a growth from day 0 to day 12. Okay, sorry, to day 10, and then it levels off to day 12. Okay, so we can conclude that there is some relationship between um, the hydrolysis of sucrose and the growth. And then all you need to do is describe your graph. There's a greater rate of hydrolysis between um, the start at 0 days up to 8 or 10 days, and then after 10 days you can see that it levels off between 10 and 12 days, and you can give a reason for that as well, because the SPS activity is leveling off. Okay, and that would be worth 3 marks. And that is it for this video. Thanks for watching. I hope you liked it. And one last thing, please subscribe, hit the like button and the notification bell.